Your brain right now is running a chemical system that evolution spent millions of years building. It's elegant, it's precise, and under normal circumstances it keeps you alive. Under modern circumstances, something has broken it. And the thing that broke it is sitting in your kitchen cabinet, your coffee cup, your morning yogurt, and almost certainly in whatever you had for lunch. Refined sugar, the kind that's been stripped down, concentrated, and engineered to hit as hard as possible, does something to your brain that looks, on a functional MRI scan, almost indistinguishable from cocaine. Same circuits, same chemical cascade, same collapse when the supply stops. And the people selling it to you have known this since the 1970s. Not suspected, known. Internal documents, archived correspondence, funded research, all of it pointing in the same direction. All of it carefully managed so it never reached you. This isn't a wellness panic. This isn't a diet blog talking point. The evidence sits in declassified corporate documents, in published neuroscience, in metabolic studies from some of the most rigorous labs in the world. When you line it all up, the biology, the history, the industry strategy, a picture emerges that is not comforting at all. If this is the kind of thing you want to understand properly, go ahead and subscribe right now. The algorithm buries this channel unless you do, and there is a lot more here worth watching. Now, let's start at the beginning, not with industry documents or addiction studies, but with something much older than either. The reason any of this worked on us in the first place. Human beings are born preferring sweet. Not learned, not acquired, born. A newborn infant, hours old, never having tasted anything processed or packaged or engineered, will show a measurable facial relaxation response when given a drop of sugar water. That same infant will grimace and pull away from something bitter. No one taught that reaction. It arrived pre-installed. The reason is ancient and makes perfect evolutionary sense. In the natural world, sweetness and danger rarely coexist. Bitter often signals poison, alkaloids, toxins, things that kill. Sour can indicate spoilage. Sweet almost always means ripe fruit, calorie-dense, safe to eat, exactly the kind of energy windfall that keeps a mammal alive through the lean season. Over millions of years of selection pressure, the mammalian nervous system built a dedicated detection system for sweetness and wired it directly to the brain's reward circuitry. Find sweet, feel good. Remember where it was, go back. Two protein subunits on the tongue, T1R2 and T1R3, couple together to form a sensor tuned precisely to sugar molecules. When they bind, they send a signal upstream so fast, it's almost instantaneous. Before you've even consciously registered the flavor, your brain is already beginning to respond. That response was never supposed to be a problem. In the ancestral environment, sweet things were rare. A patch of wild berries, a honeycomb discovered by chance. The reward the brain offered for finding them was proportionate to their scarcity. The system worked beautifully for about 200,000 years of human history. Then we figured out how to refine sugar, concentrate it, and put it in everything. We took a survival mechanism tuned for scarcity and dropped it into a world of manufactured abundance. The ancient wiring didn't change. The environment around it changed completely. That mismatch is where the damage begins. When sugar hits those taste receptors and the signal travels upstream, the first major stop is a region buried deep in the brain called the nucleus accumbens. Think of it as the brain's central reward processing node, the place where the question, "Was that worth doing again?" gets answered. The currency it uses to answer that question is dopamine. Dopamine is not, as it's often described, simply the pleasure chemical. It's more precise than that. Dopamine is the anticipation and motivation signal, the neurochemical that says, "That mattered. Pursue more of it." When it floods the nucleus accumbens after a sweet taste, it doesn't just create a sensation of pleasure, it stamps the experience into memory and builds a motivational drive to repeat it. Taste, dopamine surge, memory encoding, craving. That's the loop. Sugar doesn't stop at dopamine. The reward system connected to the nucleus accumbens also has an opioid component. Yes, the same class of receptors that respond to morphine and heroin. Specific receptors in the reward pathway respond to sweet taste by releasing the brain's own opioid-like molecules, the endorphins and enkephalins, that produce what researchers describe as hedonic pleasure, actual physical comfort. The warm glow of a dessert is real, it's physiological, and it involves the same receptor type as some of the most addictive substances on Earth. Now here's what makes this system go wrong. The dopamine surge hits hardest on the first encounter with a stimulus, then diminishes with repetition, habituation. The second bite of something never hits quite as hard as the first. The 10th hits less than the second. The brain is always calibrating. Is this new information, or is this expected? Expected rewards produce a smaller dopamine signal than surprising ones. This is the trap. The pleasure decreases, but the learned motivation, the craving, doesn't shrink at the same rate. You end up with a system that drives you towards something delivering diminishing returns. The circuitry keeps sending the signal to seek. The satisfaction keeps dropping. In the late 1990s, a neuroscientist at the University of Bordeaux named Serge Ahmed was running addiction experiments on rats.
The initial question had nothing to do with sugar.
Ahmed was studying cocaine dependence, trying to understand what makes a drug so difficult to stop taking, even when it's destroying the animal's health. The setup was standard. Rats were given intravenous cocaine through a catheter. Over time, they escalated their use, spending more of their day self-administering the drug, showing all the behavioral signatures of compulsive addiction. A validated, well-established model of substance dependence. Then, Ahmed introduced something outside the original protocol. He gave the addicted rats an alternative, a lever delivering intensely sweet water. Saccharin first, then later sucrose. The rats could choose, at any moment, between the intravenous cocaine they were already dependent on and the sweet taste. The result was not what anyone predicted. A large majority of the rats switched to the sweet option, persistently, repeatedly, even after weeks of cocaine use had already produced physiological dependence. Ahmed and his colleagues published in 2007, under a title that made the finding impossible to misread. Intense sweetness surpasses cocaine reward. The explanation Ahmed proposed was evolutionary. The T1R2 and T1R3 receptors evolved in environments where concentrated sugar simply didn't exist. They were never calibrated for the levels of sweetness that humans can now manufacture. Hit those receptors with a highly concentrated sweet stimulus, and you generate what researchers call a supranormal reward, a signal stronger than anything the evolutionary environment could produce. The brain's self-regulation mechanisms, built to handle normal inputs, get overwhelmed. The scientific community's response was cautious. The standard critique was that rat models don't map perfectly onto human neurobiology, which is true. Others argued that preference isn't the same as dependence. The paper generated significant debate, as it still does. But the core experimental result, that given the choice, a majority of cocaine-dependent rats chose sweet, has been replicated. The interpretation remains contested. The data doesn't. And it raised a question that subsequent human neuroscience spent the next decade trying to answer. If the reward signal from concentrated sweetness can outcompete cocaine in a rat's brain, what exactly is happening in a human brain that consumes engineered sweet products dozens of times a day, every day, from childhood? The answer runs through three mechanisms that together constitute the hallmarks of physical dependence: tolerance, craving, withdrawal. Each one appears clearly in the neuroscience of sugar consumption, and each is measurable in ways that go well beyond self-reporting. Tolerance develops first. The dopamine system, faced with repeated large stimulation from high-sugar inputs, responds by downregulating, physically reducing the number of active dopamine receptors in the nucleus accumbens. Fewer receptors means the same stimulus produces a weaker signal. Neuroimaging studies in human subjects maintained on sustained high-sugar diets have documented measurably reduced dopamine receptor sensitivity. The amount of sweet that satisfied you a year ago no longer produces the same response. You need more to return to baseline. The baseline itself has shifted downward. Craving intensifies in parallel. When the dopamine signal is blunted, the wanting circuitry, which neuroscientists distinguish carefully from the liking circuitry, actually amplifies. You want more, even as the pleasure of having it decreases. This is the subjective experience millions describe as being unable to stop at one biscuit, one piece of chocolate, one serving. It feels like weak willpower. Neurochemically, it's a signal that has become decoupled from the pleasure it was supposed to track. Withdrawal is the part that surprises most people, because we tend to associate it with dramatic physical symptoms, the shaking of alcohol detox, the agony of opiates. Sugar withdrawal is subtler, but physiologically real. When habitual high sugar consumption stops abruptly, brain imaging studies show measurable changes in dopamine and opioid receptor activity. Irritability, low mood, difficulty concentrating, fatigue, symptoms almost universally dismissed as psychological, but reflecting an actual neurochemical readjustment in the reward system. The brain had calibrated itself to a certain daily input. Remove it, and the system is out of balance. Those weeks of recalibration feel bad. And that discomfort is one of the most powerful drivers of continued consumption, because the fastest way to stop feeling it is the same thing that caused it. This cycle, tolerance, craving, withdrawal, consumption, temporary relief, tolerance again, is not a metaphor for addiction. It is the mechanistic definition of it. Understanding that cycle brings us directly to the question of who else understood it and when. In the summer of 1965, a Harvard nutrition researcher named D. Mark Hegsted received funding from an organization called the Sugar Research Foundation. The grant wasn't unusual by itself. Industry funding of academic research was common, accepted, and largely unquestioned. What was unusual was what Hegsted produced in exchange for it, and what the Sugar Research Foundation specifically asked him to produce. The documents revealing this arrangement sat in archival boxes at the University of Illinois for decades. Nobody looked for them because nobody knew to look. In 2016, a researcher named Kristin Kearns went through those archives and found what the industry had been paying for. The correspondence was meticulous. Project 226, as the Sugar Research Foundation internally labeled it, had a clear objective: produce a literature review that would exonerate sugar from its growing association with heart disease and redirect scientific attention toward dietary fat. Hegsted delivered. The resulting papers, published in the New England Journal of Medicine in 1967, systematically dismissed studies linking sugar to cardiovascular disease and elevated the case against fat. The reviews were presented to the scientific community as independent academic analysis. The funding was not disclosed. It would not be disclosed for nearly 50 years. What makes this history particularly pointed is what Hegsted did next. The same researcher whose work was funded and directed by the Sugar Research Foundation later became head of nutrition at the United States Department of Agriculture. He was the person who oversaw the development of America's first official dietary guidelines in 1977, and the dietary guidelines that followed in 1980. The foundation laid by Project 226 didn't stay in academic journals. It walked directly into federal nutrition policy. The man who coordinated the industry effort was a lobbyist named John Hickson. In internal memos never meant to leave the room, Hickson laid out the strategy with uncomfortable directness. The goal wasn't simply to defend sugar. It was to permanently shift the scientific conversation away from sugar as a health concern and toward fat as the dietary villain. In one documented internal communication, Hickson described the objective as a long-term program of industry-funded research designed to counter what he called negative attitudes toward sugar forming in the scientific community. He identified specific researchers whose work was inconvenient. He mapped out how continued funding could be used to ensure the literature moved in the desired direction. Hickson understood that you don't need to suppress inconvenient science. You just need to fund enough convenient science to drown it out. Within a decade, mainstream scientific consensus had moved exactly where he paid for it to move. Sugar was largely cleared, fat was the enemy, and an entire generation of nutritional science was built on a foundation that had been, from the beginning, commercially engineered. The consequences didn't stay inside academic journals. They spilled into policy, into federal dietary guidelines, into the food industry's understanding of what it could now, with scientific cover, do to its products. Walk into an American supermarket in 1985, and what you see is a transformation so thorough it looks organic. Low-fat everything. Fat-free everything. The word fat on a food label had become the equivalent of a skull and crossbones, and manufacturers had raced to remove it. Low-fat cookies, fat-free salad dressing, reduced-fat crackers that tasted, to put it plainly, like cardboard. Removing fat from processed food creates a chemical and textural problem. Fat carries flavor, provides mouthfeel, and contributes to satiety. Take it out, and you have something that neither tastes good nor keeps anyone full. The solution the food industry arrived at was obvious and catastrophic. They replaced the fat with sugar. In some cases, more sugar than had ever been in the original full-fat product. The calories stayed roughly the same, or climbed higher. The satiety effect collapsed, and the product now carried a health halo stamped across the front of the package. The timeline of what followed is not subtle. American obesity rates, relatively stable through the 1960s and early 70s, began climbing sharply in the late 70s. By the mid-90s, physicians were describing the trajectory as an epidemic. Childhood obesity rates, around 5% in 1970, had tripled by the time the decade ended. The low-fat guidelines intended to protect cardiovascular health had coincided precisely, not approximately, precisely, with the sharpest increase in obesity and type 2 diabetes ever recorded in the country's history. Dietary fat in food had dropped. Sugar content had surged. The population following the guidelines was getting sicker. The connection was visible in the data for anyone looking carefully. Most of those designing the guidelines were not looking carefully, or were funded by organizations with a strong interest in where the looking went. Howard Moskowitz does not look like a man who reshaped the American food supply. Compact, energetic, given to rapid, enthusiastic explanations, he holds a doctorate in experimental psychology from Harvard. He started his career in the early 1970s working for the U.S. Army, which had a specific and practical problem. Soldiers in the field weren't eating enough rations, even when they were hungry. His job was to figure out why. What he found was something the food industry would later consider one of the most valuable discoveries of the 20th century. The problem wasn't that the rations tasted bad, it was that they were too consistently stimulating. When soldiers ate the same intensely flavored food repeatedly, they stopped, not from fullness, but from what Moskowitz termed sensory-specific satiety. The brain's response to a consistent stimulus diminishes over time. A food engineered to a single flavor peak causes consumption to drop precisely because it's too good at what it does. The solution Moskowitz described in his papers, and later in commercial work, was finding what he called the bliss point. Not maximum sweetness, or maximum saltiness, or maximum fat concentration, but the precise combination of all three that produced the highest drive to keep eating without triggering the satiety response that stops consumption. The bliss point isn't the most pleasurable single bite. It's the concentration that prevents the eater from stopping. Moskowitz brought this mathematical framework to Pepsi-Cola in the mid-70s, working on their Diet Pepsi formula. The method worked well enough that he spent the following decades consulting for companies including Campbell Soup, General Foods, and Kraft, organizations whose products were eaten by hundreds of millions of people every day. Product development teams across the food industry began running bliss point optimization studies systematically, feeding thousands of test subjects incremental variations in sweetness and measuring not preference ratings, but consumption rate. Preference tells you what people say they like. Consumption rate tells you what they actually keep eating. Those two numbers are often very different. Every time you open a package of something processed and find you've eaten the whole thing before registering what happened, you have encountered the output of this methodology. It isn't an accident of design, it's the intended result. Not all sugars behave the same way once they're inside you. For most of the low-fat era, the food industry and much of the nutritional establishment treated a calorie as a calorie and a sugar as a sugar. The biochemistry tells a different story. Table sugar, sucrose, splits in the digestive system into two parts: glucose and fructose. Glucose enters the bloodstream and triggers the body's standard metabolic response. Insulin is released. Cells take up the glucose. The hormone leptin signals the brain that energy has arrived. Satiety begins. The process runs as designed. Fructose doesn't take that route. The liver captures it almost entirely before it reaches general circulation. Unlike glucose, fructose doesn't stimulate meaningful insulin secretion. Without that insulin signal, leptin isn't adequately triggered. The brain never receives a clear message that enough energy has arrived. You can consume a substantial number of fructose calories and remain, neurochemically speaking, in a pre-fed state. Inside the liver, fructose is metabolized through pathways that generate uric acid and drive fat production, a process called de novo lipogenesis, the liver manufacturing new fat from the incoming molecular substrate. Robert Lustig at the University of California, San Francisco, drawing on detailed clinical work, has described this metabolic signature as nearly identical to what happens in the liver during chronic alcohol consumption. Same enzymatic pathways, same fat accumulation mechanism, different social acceptability. A pivotal clinical demonstration came from a study published in 2009 in the Journal of Clinical Investigation. Two groups of overweight subjects consumed identical total calories over 10 weeks. One group's supplemental calories came from glucose, the others from fructose. Both groups gained weight. Only the fructose group showed increases in visceral abdominal fat, the metabolically dangerous kind packed around internal organs, along with elevated triglycerides, decreased insulin sensitivity, and markers consistent with early metabolic syndrome. Same calories, dramatically different outcomes. The molecular route the sugar took inside the body made all the difference. High fructose corn syrup, which became the dominant sweetener in American processed food and beverages through the 1980s because it was cheaper than sucrose, is approximately 55% fructose. The industry understood this. For most of those decades, the public did not. The satiety system, under normal conditions, works like a thermostat. As body fat increases, fat cells release a hormone called leptin into the bloodstream. Leptin travels to the hypothalamus, which reads the signal and suppresses appetite. Energy stores are sufficient. Stop eating. In a healthy metabolic state, the calibration is remarkably precise. Chronic high fructose consumption breaks the thermostat. Specifically, it produces a condition called leptin resistance, in which the hypothalamus stops responding normally to leptin's signal. The hormone is still being produced, in people with obesity, often at elevated levels, but the brain has effectively stopped registering it. The signal says, "Stop eating." Nothing happens. Simultaneously, the hunger-driving hormone ghrelin is not suppressed by fructose consumption the way it is by other nutrients. A meal deriving significant calories from fructose-rich beverages or processed foods leaves ghrelin elevated longer than an equivalent meal composed differently. The hunger signal keeps running even after the calories have arrived. Clinical observations published in the early 2000s documented patients with significant obesity reporting genuine difficulty feeling full at any normal meal size. When researchers measured their leptin levels, the hormone was present at 5 to 10 times normal concentration. The brain had simply stopped registering it. Eating past any rational endpoint, with the biological signal that should extinguish hunger no longer getting through, this is not a willpower story. The feedback loop had been disrupted at the level of receptor sensitivity. If this is what sustained sugar exposure does to a fully formed adult brain, with all its regulatory systems in place, the question that follows is an uncomfortable one. What happens when the same exposure begins not in adulthood, but in the first years of life, when the reward system is still assembling itself, when the dopamine pathways are still being calibrated, when the entire architecture of wanting and satisfaction is being built for the very first time? That question was studied. The results were known long before the public heard the words fructose or leptin resistance, and what the industry did with those results is its own chapter entirely. There is a document, not classified, not particularly dramatic looking, produced by a market research firm in the United States in the early 1950s. It was commissioned by several major food and beverage companies who shared a question they needed answered. At what age does brand loyalty form in a child? The answer the researchers returned was startling enough to shape the next 7 decades of food marketing strategy. Brand and taste preferences, they found, could be established as early as age 2. Not age 12, not age 8, age 2. The window in which a child's flavor preferences and emotional associations with food were being permanently encoded was far earlier than anyone had commercially appreciated. Of the stimuli producing the strongest and most durable preferences, intense sweetness ranked highest.
A child given sweet foods repeatedly in the first years of life didn't just like sweet foods. The child's reward system, still under construction, calibrated itself around that input. The preference wasn't acquired the way you acquire a language. It was embedded at the level of neurological architecture. The executives who received this research didn't file it away. They built product lines around it. Breakfast cereals aimed at children began appearing in the late 1950s, with sugar concentrations that would have seemed excessive even by adult dessert standards. Some containing more than 50% sugar by weight. Packaging featured cartoon characters, bright primary colors, prizes inside the box. Every element was designed for a person between 2 and 10 years old. The cereals were placed on lower shelves so children could reach them. Television advertising during Saturday morning cartoons became one of the most concentrated advertising environments in the country's history. By the early 1970s, the average American child was seeing more than 20,000 food advertisements per year, the overwhelming majority promoting products high in sugar. The developing brain is not simply a smaller adult brain. The prefrontal cortex, responsible for impulse control, delayed gratification, evaluating consequences, is not fully developed until the mid-20s. In a young child, it barely functions in this capacity. What is fully operational and highly responsive is the reward circuitry. The dopamine system in a child's brain responds to pleasurable stimuli with greater intensity than in adults, and the receptor downregulation that comes with repeated overstimulation happens faster. A child's reward system, repeatedly flooded with the supranormal sweet signal Ahmed's rats had demonstrated could outcompete cocaine, calibrates its baseline around that stimulation level. By the time that child is old enough to make independent food choices, the reward system has spent years being trained to expect a level of sweetness that no natural food can provide. In the spring of 1998, the San Francisco Unified School District was facing a budget shortfall that was, by the standards of underfunded urban school systems, not unusual, but genuinely severe. Programs had been cut. Staff had been reduced. Into this environment came a proposal that districts across the country were already accepting. The arrangement was called a pouring rights contract. In exchange for exclusive placement of a beverage company's products in every school vending machine, cafeteria, and sports facility, the company paid the district a licensing fee, guaranteed annual income continuing as long as the products were sold. Similar deals were being signed across the United States through the 90s. By the early 2000s, the majority of American high schools had exclusive agreements with major soft drink manufacturers. These contracts typically included volume incentives. The more product the school sold, the larger the payment. A district administrator sitting across from that proposal understood, without anyone saying it directly, that income would increase if consumption increased. Vending machines appeared in hallways. Sports drinks were available at lunch. Some agreements specified placement near gymnasium exits, where students would pass after physical activity. A school board official in Colorado Springs sent a memo to district principals in 1997, later reported in the press, instructing them to encourage students to consume soft drinks during the school day, in classrooms, not just at lunch, in order to meet volume targets. When the memo became public,
The district faced criticism. The contract stayed in place. The phrase that appeared in district after district when these arrangements were later scrutinized was always some variation of the same sentence. The money was too important to refuse. Not that the contracts served students, that the money couldn't be turned down. In 2009, a professor of pediatric endocrinology at the University of California, San Francisco, posted a lecture to YouTube. His name was Robert Lustig, and the lecture was called Sugar, the Bitter Truth. It ran 90 minutes. In format, it was a standard academic seminar. A researcher with a whiteboard and clinical data, working through the metabolic mechanisms of fructose for what was originally a medical education audience. It spread in a way that almost no academic lecture ever has. Within a couple of years, it had accumulated millions of views, an extraordinary number for 90 minutes of university biochemistry. Something in what Lustig was saying broke through in a way that decades of dietary advice had not. What he presented wasn't new to researchers in his field. The mechanisms he described had been published in peer-reviewed literature throughout the 2000s. What was different was the directness. Lustig named the substance, described the mechanism, showed the clinical data, and said plainly that chronic fructose exposure at current consumption levels was causing harm at a population scale. He used the word toxic. He said the food industry knew this and had structured its products accordingly. The response from food industry trade organizations was immediate and forceful. The Corn Refiners Association, representing manufacturers of high fructose corn syrup, launched a substantial public relations campaign, emphasizing that all sugars were metabolically equivalent. Researchers with industry funding published letters and op-eds questioning Lustig's conclusions. The phrase, "a calorie is a calorie," appeared in counter-messaging with a frequency that suggested coordination rather than coincidence. Meanwhile, the lecture kept spreading. Primary care physicians watched it and began having different conversations with patients. Science journalists covered it. People who had struggled with food for years watched 90 minutes of biochemistry and felt, for the first time, that what they'd experienced had a physiological explanation, a mechanism in the body, rather than a failing in character. That shift in framing, from moral weakness to neuroscience, was the part the industry had most reason to fear. The people who have stopped eating sugar, not reduced it, not swapped it for artificial alternatives, but eliminated it as completely as modern food labeling allows, describe the first 2 weeks in terms that cluster around a narrow set of experiences: headaches, fatigue disproportionate to any obvious cause, irritability arriving without clear triggers, a specific, insistent craving with a physical quality distinct from ordinary hunger, located somewhere between the throat and the chest, difficulty concentrating, low mood that lifts, usually around day 10 to 14, with a clarity that feels surprising, as if something normalized as baseline had actually been a form of dampening. These accounts are not merely subjective. A 2013 study, published in the European Journal of Nutrition, examined brain imaging data from subjects undergoing significant dietary sugar reduction. The scans showed measurable changes in dopamine receptor availability in the nucleus accumbens, the same region implicated in substance dependence, the same receptor population that downregulates under chronic oversaturation. As high sugar input was withdrawn, receptor density began recovering. The brain, released from sustained overstimulation, was recalibrating toward the sensitivity it had possessed before the habituation set in. Animal model research has documented the neurochemical signature of sugar withdrawal with particular precision. Animals maintained on high sugar diets and then deprived showed measurable drops in dopamine release in the nucleus accumbens, anxiety-like behavior, and, when given renewed access, binge patterns consistent with relapse in dependency models. At every measurable point, the system behaved like a system in withdrawal from a substance that had produced genuine physiological dependence. For the people who move through those 2 weeks and come out the other side, the food landscape shifts. Fruit, which had tasted unremarkable against the backdrop of engineered sweetness, begins tasting intensely sweet. Foods that had been neutrally acceptable become satisfying in a way that feels qualitatively different. Not the urgent, forward-leaning drive of a dopamine-mediated craving, but something slower and more complete. The reward system, no longer flooded daily past its natural threshold, has returned to a calibration where ordinary inputs produce ordinary responses.
The ancient wiring, evolved for a world of scarcity, is finally running in the conditions it was built for. That recalibration is real, it's measurable, and it points directly at a question with no comfortable answer. We built a food system that spent 50 years optimizing one variable: consumption. Not nourishment, not satiety, not long-term health. How much, how often, how early in life the habit could be established, and how difficult it could be made to stop. The science that might have constrained this was redirected by the organizations that funded it. The children most neurologically vulnerable to this targeting were the ones most aggressively marketed to. The regulatory systems meant to protect public health were slower, less funded, and less coordinated than the industry working to outpace them. The substance at the center of all of this carries no warning label. It appears in infant formula, baby food, school lunches, hospital cafeterias, and the majority of packaged products on every supermarket shelf in the developed world. Current estimates from health monitoring surveys suggest the average American adult consumes somewhere between 15 and 20 teaspoons of added sugar per day, well above what leading health organizations recommend. Children, in most surveyed populations, consume more. The neuroscience isn't ambiguous. The reward pathway activated by concentrated sugar is the same pathway activated by cocaine and heroin. The mechanisms of tolerance, craving, and withdrawal map onto established dependency models with a precision that has survived rigorous peer review. The industrial documentation proving that this was known, managed, and strategically obscured is archived correspondence, now public record, cited in academic literature and taught at universities. None of this means eating a piece of cake is morally equivalent to drug use. That's not what the science says. What the science says is that a product was engineered to exploit a specific vulnerability in human neurobiology, that the exploitation was deliberate and documented, and that the consequences have played out in the bodies and medical histories of hundreds of millions of people who never had access to the information required to make a genuinely informed choice. There is a version of this story where the worst is behind us, where the documents are public, the mechanisms are mapped, the industry is reforming, and the knowledge we've accumulated is enough to reverse the trajectory. The data on sugar consumption in children, and on the rate at which conditions once rare in adolescents are now appearing in pediatric clinics, suggests we are not yet living in that version. We are left with a system designed to be difficult to exit, applied most intensively to those least equipped to resist it, sustained for decades by the careful management of a scientific conversation that was never supposed to reach us. The mechanisms are visible now. The question of what we do with that visibility is still open, and more urgent than it has ever been. If this is the kind of story you think more people should hear, share this video and subscribe. There's a lot more on this channel worth your time, and the only way it finds people is if you help it get there.